fertilization, the beginning of pregnancy on this discussion. And I want to continue some of that. Before I do, there was a question that was just raised I want to clarify. Every year I assign 3 optional readings, which are books, usually in the popular press, that I suggest that you read during the term when you're not really in the mood to study deeply the material in class. What I'm trying to do is to connect what goes on here to what goes on in the world around us. And also allow you to see how endocrinology shows up in various people's lives in various places. Over the years, we've done probably 30 different titles for books. And obviously, when I put my syllabus together, sometimes I make mistakes and I forget or to make changes. The 3 books this year are correctly listed in the syllabus. One of them is called Moonlight Sonata. At the Mayo Clinic. And that one is about somebody who comes up with a diagnosis that apparently has some relationship to the endocrine system, but you're not sure what. And so it's a mystery, it's kind of a mystery story. Okay. And it can plug into a variety of places, including the segment on cancer during the term. There's another one. See if I can remember the title. Actually, you've got the titles.
In due time.
In Due Time. In Due Time is a biographical story about fertility treatment. Which obviously fits together with some of the things we've been talking about in class of late. And the other one is a classic book by Cannon, who was at Harvard. And it's about bodily changes, and it's a long, long title. It was originally published in 1925. And it's a reprint of papers that describe the adrenal function. And part of the reason I assigned that was to have people understand a little bit about how endocrine subjects progress over time. And also to let you know that even in 1925, people were able to figure out a lot of what actually was going on in the body, even without, gene splicing and knockout animals and many of the new techniques we have now, but using old style basic physiology and a lot of deductive reasoning. So these were the 3 that were assigned as optional reading. You don't have to do any of it. You're going to get potential credit of up to 40 points on the final exam for answering a series, I think it's of 8 questions. 8 times 5 is 40. 8 questions on the final exam that deal with each of these books. And those questions obviously are essentially trying to figure out what you whether you, number one, read them, and number two, what you drew from them. Yeah?
Is this 40 out of 100?
Pardon?
Is this 40 points out of 100 points?
No, the final exam is 200 points, basic 200 points. You can get up to 240 if you got everything right on the exam, plus 40 points on extra credit. So it's a nice chunk to get if you don't just write down the title and the author, because you won't get many credit points for that. There was another question back here.
We'll get some points from writing down the title now.
Maybe. Depends on how I write the questions. Okay. Okay, so that's the extra credit reading. And I think the longest of these is about 250 pages. Large type anyway. So they're short. Okay. Now, we showed you how fertilization occurred. How does a woman know she's pregnant? Pregnancy test. Naturally, maybe I should put in there. How does she naturally know that she's... Okay, missed period. So she's not menstruating when she thought she would be menstruating. It's going to come... Anything else? Nausea. That actually comes a little later. But it does play a role.
Doesn't she usually feel tired?
Fatigue is a tough one because I feel tired when I get up in the morning sometimes. And I'm not pregnant. Anything else?
Starts getting bigger.
The first day.
Believe me, you would be surprised what a 6-month pregnant woman looks like, particularly if she's been training. Okay? You won't be able to determine that she's pregnant. Okay, so a little bit about some of those questions. Some of those questions actually get back to things we've gone over before. And it involves the hypothalamus, the pituitary, the hormones that are produced here, their effects on the corpus luteum, and the progesterone's action both centrally and on the endometrium. Okay. This is in the non-pregnant state, maternal, we're talking about maternal recognition of pregnancy. So in the non-pregnant state, you've got a cycle that looks something like this. Let me see if I can get this to shrink a little. Down. Okay. So we know all about, where does the LH come from? What cells do we get that from? Gonadotropes, which are located where?
Anterior pituitary.
Anterior pituitary, okay. What about prolactin?
Corpus luteum.
No. Where does it come from?
Oh, wait. That was progesterone. My bad. Prolactin comes from, wait.
Okay, and it comes from the pituitary, but what cell type in the pituitary? Lactotropes or mammotropes, okay? So those two hormones. Prolactin plays a rather minor role in primate corporal luteal function, but it plays a big role in a lot of other species. That's why it's up here. Those stimulate the corpus luteum to form progesterone. And that progesterone goes into circulation, it acts to differentiate the endometrium. It modulates estrogen's actions just about everywhere. Estrogen action in the breast, for instance. One of the things it does is stimulate the formation of progesterone receptors. Progesterone then acts. And it's a negative feedback regulator at the level of the hypothalamus. Kisspeptin, neurotransmitters, LHRH production. Okay, so it's negative feedback. Notice, however, that in between here, progesterone acting on a non-pregnant uterine endometrium stimulates the production of prostaglandins, particularly prostaglandin F2 alpha. And that prostaglandin gets back to the corpus luteum via a system that we call I'll show you the vascular system, that cross-current transfer back to the arterial supply for the ovary, where it suppresses luteal growth and luteal continuity. So recognize that without a pregnancy here, this can occur for a period of time. This is shutting down LH production and prolactin production. This is actively causing apoptosis in luteal tissue. So you've got essentially a suicide loop for the corpus luteum. More progesterone, more prostaglandin, more progesterone, more prostaglandin. And that's going to terminate the activity of the corpus luteum. Now, in a non-pregnant primate ovary, how long did I say that the corpus luteum normally lasted?
14.
Pardon?
14 days?
14 is a little long.
10 plus or minus
10 plus or minus 2 days. Okay? So you've got something around 10 days worth of corporal luteal function in the presence of this suicide loop. By the end of that period of time, you're getting to the point of induction of menses, loss of the endometrial lining, breakdown of the spiral arterioles, loss of blood, the bleeding that signals this next cycle. Okay, and the corpus luteum is going on to form a corpus albicans, just a scar effectively. So, what comes next? What about in the case where we get a pregnancy? Well, actually, let's see if we can find that cross-current circulation. Oh, perfect. This was originally described in a sheep. Sheep are ruminants. They have a bicornate uterus, a two-horned uterus, and the uteri form a Y and come down a common vaginal opening. At the top end, you'll have the fallopian tubes connecting or overlying the ovaries, and the ovaries will have developing follicles as well as the corpus luteum. They are vascularized by the uterovarian artery, which goes from central circulation up toward the ovary, and they're drained by the uterovarian vein, which carries blood in the opposite direction. There are a lot of capillaries and small venules that occur along the course of these vessels that serve to also vascularize the uterus. Okay. So you've got the same blood supply and the blood drainage along the uterus, and this occurs in other species as well. Even in the human or primate, these blood vessels lie right next to the uterus, and they do vascularize the ovaries. Now, so you've got blood flowing into the ovary, and the corpus luteum, any steroids that are being produced and draining into the venous drainage are coming down the uterovarian vein, passing by the uterus, and then into central circulation. During their passage, progesterone being a steroid, prostaglandins also being lipophilic, can diffuse away from the venous drainage and into the surrounding tissue. So you can get prostaglandins from the endometrium entering the arterial supply, progesterone from the corpus luteum entering the venous drainage, and the prostaglandin gets to the blood supply of the corpus luteum, progesterone gets to the blood supply of the uterus and the endometrium. So you have cross-current exchanges occurring. Okay, that's why the messages can get back and forth. They do not have to pass into central circulation for these two messages to be exchanged between the uterus and the corpus luteum. That's important because the prostaglandins are chemically labile, and they get broken down pretty quickly. Oop, what happened? Okay. No. Where did my image go? That's important because the prostaglandin is labile. If it had to go through central circulation before it went to the ovary, it would more likely be broken down. Okay. So let's go back to what happens then in the case of the fertilization having taken place. So under these circumstances, got the same kind of a loop occurring. Here's the same loop you were looking at with progesterone. Superimposed on it, however, is something else. And that is that as a conceptus begins to interact with the endometrium, it blocks endometrial production of prostaglandins. What do you think that will do to the lifespan of the corpus luteum? Should extend it, and in fact it does. In addition, the conceptus in at least the later stages of the blastocyst and the trophoblast, which interacts with the uterus, uterine wall, you're beginning to produce, in primates at least, chorionic gonadotropin and placental lactogens. This is the placental form of LH, the placental form of prolactin. And these are getting into circulation and affecting the corpus luteum. These are non-labile chemically. They have long half-lives in circulation and can reach the corpus luteum even though they don't diffuse out of that venous drainage of the uterus. And in fact have to come full cycle through the circulatory system to get back to the corpus luteum. So you've got a removal of an inhibitory factor and the imposition of two stimulatory factors which are substituting for LH and prolactin. Why do you need this in addition to that? What's progesterone normally doing? As it goes back to the hypothalamus, it's inhibiting what? It's inhibiting LH and prolactin production. And it continues to do that. So it's suppressing centrally produced hormone, and you're getting a substitution of placentally derived hormone to help support corpora lutea development. Now it's intriguing, in other species, a lot of other species beyond primates, you will get placental lactogens formed, but you won't get chorionic gonadotropin. There aren't a whole lot of species that make a chorionic gonadotropin. Ruminants don't, for instance. Ruminants, interestingly, have supplementary corpora lutea that form about the time of conception. And those help support the endometrium differentiation development during pregnancy. Okay, so part of the recognition of pregnancy by the mother is totally unconscious as far as she's concerned. And it has to do with this switch over between central support of the corpus luteum and support of the corpus luteum by products of the conceptus and the pregnant uterus, in combination with a blockade of what is normally a breakdown inducing signal from the endometrium. Okay. All right. Now, what else is going on? Other things that are going on. Conception is out here. Notice what's going on with progesterone. It continues to be made, and most of what's being made this very early on is coming from the corpus luteum. So that continued function of the corpus luteum supports this production of progesterone. Does anybody remember when we talked about steroidogenesis, where a lot of progesterone is made in a pregnant woman? I gave you a picture that showed the maternal compartment and the placenta and the internal organs of the fetus. Does anybody remember that picture? Other than in a nightmare? Okay. It's a combined effort, effectively, to make progesterone. A lot of it starts to be made by the pregnant placenta, however. So steroidogenesis is switching from corpus luteum support to the placental production to make all this progesterone, which after a while just follows the growth of the placenta in terms of the mass that's being put into circulation. Chorionic gonadotropin from the late stages of the blastocyst, the implanting embryo, and finally the placenta proper is following this other curve. It's vastly higher than what you'd see in a non-pregnant individual, which is essentially 0, all the way through pregnancy, but it does dip after about the first trimester, so it comes down a bit and then falls abruptly at the end of pregnancy after birth. In addition, you're seeing placental lactogen going up with the weight of the placenta, as well as this various steroids, estrogenic steroids, I should say, also coming from the placenta. So all these hormones are going on. The reason mom gets nauseated early on is this and this, okay? The rapid rise in those two particular hormones. Some women get nauseated during the luteal phase of their cycle. Okay? And that's from a rapid rise in progesterone. Part of the reason being progesterone is promiscuous in the receptors it can occupy. Does anybody have a guess as to what receptors might be occupied that could induce some sort of a nauseous response? Any guesses? Yeah. Could be receptors in the digestive, but what receptors would they be? Would they be progesterone receptors? No, I said that the hormone was promiscuous. What other hormones might be associated with stimulating nausea? Pardon?
I said HCG.
No. No. Progesterone is a steroid,
Estrogen.
not estrogen. Structurally, what does progesterone resemble? It's a C21 steroid. Estrogens are C18, androgens are C19. What else is C21? See? Corticoids. Corticoids like what? Like cortisol. What's cortisol receptors associated with? Stress response, okay? Stress response. Is nausea part of the stress response?
Yes.
Are you just saying that?
Yeah.
Or is there some suggestion that maybe it is? Okay, what happens when you get an infection, temperature goes up?
Yeah, temperature goes up for enzymes work better, and also to hurt and nausea induces sort of hurt to the body of the offending substance.
Yeah, that's part of it. But the glucocorticoids are part of that temperature-induced feeling of nausea. So if you get progesterone levels that are high enough, you can get crossover to stimulate glucocorticoid type responses, including nausea. The other good thing that's going on, HCG. HCG, what's the closest analog to HCG in terms of an endogenously produced hormone? It's actually on that last slide. LH, luteinizing hormone. They both have the same alpha subunit, and the only difference in the beta subunit is what, folks, from the lab?
The long carbohydrate.
Okay, the long carbohydrate decorated beta tail, carboxy tail. Okay. So it's like LH. But if it's like LH, and what else is it potentially like? It's like FSH, the other gonadotropin, what other hormone? What? TSH. Okay. What does TSH do? Stimulates the thyroid. Okay? And what did I tell you about those various hormones if you get overproduction of those hormones? Are they pure in the sense of what receptors they stimulate? No, there can be crossover. And that's particularly true when you get really high levels, which you do with chorionic gonadotropin. We know this occupies LH receptors in the corpus luteum to keep the corpus luteum running, but it's also capable of partially occupying thyroid-stimulating hormone receptors. It stimulates the thyroid. What does the thyroid make? Thyroxine, T3 and T4. And what does that do to metabolism? Tends to speed things up, okay? It elevates body temperature. It'll do a variety of things. But part of the nausea is this very rapid rise, particularly here, in a hormone that can cross-react and stimulate other systems besides just the gonadal one. So mom's nausea has to do with steroid changes. You're also seeing in primates a pretty good bump in the levels of estrogens. Okay. Estrogens also have their own effects on central metabolism. So you've got lots of hormonal changes occurring, and you see very high levels of hormones that often aren't present in the non-pregnant state. Remember I said earlier on that we've got males and females and pregnant females in terms of physiologic states? Okay, this is in large part due to the full-blown endocrinological changes that accompany pregnancy. Okay. This is about the time, somewhere around 4 to 6 weeks, when women who have not taken a pregnancy test will show up at a clinic, okay, and be diagnosed as pregnant, because this is the point when you can start to see clearly using ultrasound that in fact a conceptus is present and a growing embryo is in the uterus. The other thing that will happen somewhere in this range is that there's a change in the sensitivity of breast tissue. Breast tissue will become much more sensitive under the influence of high progesterone and high estradiol. Okay, let's go back. All right. So, mom's recognition of pregnancy is physiological at first and biochemical. You can pick it up by looking at blood samples, at urine samples, even before mom is necessarily going to detect the fact that she's pregnant. But once you get into that, late into that first month, that first trimester, you're going to have physiological changes in mom that makes her very well aware that she's pregnant. There are plenty of individuals who will go through a lot of pregnancy and say that they never sensed that they were pregnant. It's particularly true in grossly obese individuals, where you get the big splash in the newspaper, particularly the ones near the grocery counter shelves. Okay, the Globe. What's the Globe and the? You never read those? Oh, they're great. Alien abductions, etc., things like that.
You read those?
Pardon? Well, I'm standing waiting to pay for my groceries, yes. But there's where you will see the reports on, oh, I was, woman delivered 10-pound baby and never knew she was pregnant. Okay. I had to take this with a little bit of a grain of salt. All right, let's go over here. So, what's going on with respect to that conceptus during these early stages of pregnancy? Very early on, this is just after fertilization, it's probably within 16 to 24 hours after fertilization, you get the first division and you form blastomeres. Okay. And those blastomeres are going to be present in the, for a 2-cell embryo. They're going to still be within the zona pellucida. The granulosa cells that form the cumulus would have been lost by about this stage. But the polar bodies from the unfertilized oocyte are still present. They will gradually degenerate. Every 16 to 24 hours thereafter, you're going to get divisions. And these divisions, while the embryo is still within the fallopian tube and initially in the uterus, make the cells just smaller. They aren't being enlarged. They have no source of nutrients other than the material that was contained in the original egg. The nutrients in the fallopian tube fluid or initially in the uterine fluid really aren't that rich in the amino acids, sugars, etc. that are necessary to support growth of these cells. So these guys are going through cell cycles on a regular basis, but the cells are getting smaller and smaller. In most species, the first few divisions, if you were to harvest these by going into the fallopian tube and pulling out the fertilized eggs, fertilized early stage embryos, you can take these out, break open the zona pellucida, and disaggregate these collections of cells. And what's true is that if you were then to take these and put them back into, say, an empty zona pellucida and pop them into a suitably primed mother of the same species, you could get clonal embryos. You could get embryos that had exactly the same genetic makeup because they were, in fact, the offspring or the outcomes of development of each of these blastomeres or monomeres. Genetic changes occur during these first few stages as well. Depending on which species it is, paternal genes start to be expressed in anywhere from the 4-cell to about the 16-cell stage. I believe in humans, it's between the 4 and 8-cell stage. In sheep, it's around, let's see, sheep, I think it was the 8-cell stage. Mice, it's earlier. It's actually between 2 and 4. But paternally derived proteins from the expression of the genes, the alleles on the paternal genetic contribution to the embryo, only starting to show up usually well after the 2-cell stage, and sometimes after stage 4 or even 8. So those come in later. Once you get down to about this stage, you're getting to the point where the embryo is moving into the uterus. And you're getting what's called hatching. The zona pellucida is starting to thin and or disaggregate. you've got the early blastocyst with the blastocystic cavity, most of the cells contributing to the outer layer of the blastocyst. These are going to go on and form the trophoblast outer layer, and they are going to form the placenta. It's only a minor component of the cells that you started with that are actually going to make the embryo proper. So this is the late blastocyst. This is about the stage when you're going to start interacting with the uterine endometrium and actually getting into the vascular maternal cell wall, embryo or uterine wall. There are a couple of slides here that show the various stages of this process called implantation, or more technically, nidation. These are largely taken from Moore's The Developing Human, which is kind of the Bible for development of the embryo in the human. And here is the slice through the endometrium. Notice it's pretty thick. There is a more differentiated layer of cells on the surface, the epithelium. There's a lot of vascularity within this tissue. There are these spiral, these pits that contain a lot of glycogen in the cells that surround them. There is, before contact is made, there's a lot of endocrine chemical crosstalk between the maternal component and the embryonic component. I talked to you about some of that with respect to insulin-like growth factor 1, which is made by the blastocystic cells, particularly the cells along this face of the embryo. You're making IGF-1 in the maternal endometrium. Maternal endometrium is making a binding protein for IGF-1. In addition to IGF-1, the blastocyst is making a proteolytic enzyme that breaks down the binding protein for IGF-1. And you've got an inhibitor of that proteolytic enzyme made by the endometrium. So there's this back and forth, and it's a balance between that crosstalk, that chemical crosstalk, not just for IGF-1, but for several other proteins. One is called LIF, L-I-F, as well, that occurs across this space and ends up determining where on the endometrial wall the embryo is finally going to make contact and begin invading. And that's indeed what it does. These cells at this face form what's called the syncytial trophoblast. The syncytial trophoblast is a rich source of HCG. And these cells tend to form giant cells. Syncytial refers to a structure in which multiple nuclei occupy the same cytoplasm. So the heart is actually a syncytium because the cells have open channels, cytoplasmic channels, between the various smooth muscle cells of the heart. It beats in unison. You run into that in the smooth muscles of the uterus at the time of delivery as well. So this is syncytium. It's making proteolytic enzymes that help break down the intracellular proteins and allow it to invade the maternal endometrium. While this is going on, you've got cell division going on in these placental or cytotrophoblastic cells that are going to form the placenta, as well as in the inner cell mass. This is all going on, again, prior to the time when the mother would normally be just approaching the time of menses, subsequent menses, if she had a non-pregnant cycle. As this continues, this syncytiotrophoblastic tissue continues to enlarge and invade. You're getting more differentiation in the inner cell mass to form multiple layers of cells. The beginnings of the formation of the amniotic fluid cavity. This blastocystic cavity is going to get smaller over time. You start to form multiple layers of the placenta as well, which are continuing to invade. And notice that these tissues are getting closer and closer to maternal circulation. This is just this blown up, showing you these various cell types. And then finally, in primates, you get a complete invasion of the embryo into the maternal endometrial cell wall, and there is a closure over the actual conceptus. So here's the endometrial surface. So the lumen of the uterus would be out here. And you get closure. So this is actually invaded into the endometrium. It hasn't gotten to the myometrium, the muscle layer that lies down here, and the outer portion of the uterus, which would be up here. But it is getting into contact with blood vessels. Okay. This is where as this is approaching, this is where a lot of HCG is starting to get into the blood vessels in the mother, and it's showing up in serum and in urine at that stage. So trophoblastic recognition, adherence to endometrial epithelium with stimulation of this decidual reaction, final differentiation, and the beginning of trophoblastic invasion is what you call nidation. Okay, so next time we're going to talk about some things that occur during pregnancy and get into the discussion of sex differentiation. Folks in lab, don't forget your 3 lab report drafts tomorrow, and we get started on the last lab segment.
We're great on